Hello, my name is Haley DeLong, and I'm going to be doing a presentation on the anatomy and physiology of the retina. Here is my paper and the date. Today is May 12th, 2022. Okay. Not sure if you can still see me, so I'm just going to leave it like this. Okay, so first off, this is the title slide, and my name is down there on the right side. In the first slide, I'm going to be just discussing a little bit about the anatomy of the eye. So here you can see the anatomy. So first off, the light is going to enter the eye and essentially goes through the cornea and the lens, also through the pupil, which I'll talk about a little bit later. But through the lens, it kind of focuses the light on the retina, which is this yellow type of line that surrounds the eyeball. The sclera kind of goes around that as well, which is more so like a protective layer that kind of reinforces the area and creates the shape of the eyeball as well. So the pupils, basically, you've seen in your own eyes when you open your eyes, your pupils are dilated, but when after a few minutes, they kind of go back to a normal size. So those relax and dilate based on light levels and levels from your sympathetic and parasympathetic nervous system, which we learned about in this unit as well. So the cornea, which you see here, it's like the outermost outer layer, and the lens both work together to take the light rays and bend them so that they can be processed in the retinal layer. And then the second picture here on the right side is going to be a more in-depth layer. So this little square box that's here shows on the right side is going to, like on the further right side, is going to be the more like in-depth version of it. In further slides, I'll go into this a little bit more, but this is like a generalized photo of what the retinal layers look like. So this one would be the one that's closest to like all of these blood vessels and all that. And all of these layers work together in conjunction to, the light goes through them and then the signals go out back into the brain so that you can process each thing. And I'll go over the physiology later. So I like this picture because it demonstrates the pathway of the light and the directionality of the signal. So you can see that down here on the bottom left. So the light goes from the axons of the ganglion cells all the way to the pigmented layer, and then the signal goes the opposite way. So the signal output, basically the action potentials that form those signals that we will be able to process in our brain, go the other way. So they kind of cross each other like this. And then this shows like the more in-depth layer of what the retina looks like. And then on the right side here is the same picture, but they show it like on a cellular level, what it actually looks like. So first of all, you're going to see that the light comes through the axons, but it kind of bypasses through these cells, and then they go more so of the light is going to go through the photoreceptors, and that's where most of it is kind of captured. And then the pigmented layer kind of acts like a safety net almost for the rest of the light that might escape from the photoreceptors. So it kind of acts like that, so to protect the light and all the photoreceptors kind of as well. And then the choroid here, you can see on the right side with like the dark splotches through it. That's like full of blood vessels, and that supplies nutrients and oxygen and all this stuff that keeps the process going all the time so that we can continue to see. And then I'll go into more about how everything is kind of captured and processed through these photoreceptors more so in the next few slides as well. But here is the names of all the cells that are used, and I'll go over that more as well as we continue. So more on the photoreceptors. So here is more so about the rods. So the rods are going to function better in low light. And then if you were to watch like a black and white TV show, this is what you would primarily use. So it's easier on the eyes, and it uses you're using that more so at nighttime. So then we also have the cones. Those are going to function with more light during the daytime, color vision. So when you watch a color TV show, the cones are going to be more active. So here, this picture shows like an in-depth version of like this part here that I am circling. I'm not sure if you could see my cursor or not. But the pigment epithelium is at the top here. So that's going to show that how that extra light is kind of reflected back. But down here, you can see where the little light arrow is, the bottom. So that's going to be the flow of light toward the dark pigmented epithelium. So here you can see the discs as well, which I will talk about a little bit. But yeah, basically in this section here, I just wanted to mention what the rods and cones were and what kind of lights they worked best in. And I'll talk about a little bit more about the synaptic terminals that we kind of talked about in the first few units of the first few lectures of this unit, and how they synapse with the bipolar cells, which in turn causes an action potential. I'll talk about that more too. But here is also a picture that just keep noted, because I'll talk about that in a few minutes too, about the discs and then the rhodopsin molecules. So basically, phototransduction is going to be the process in which humans or animals convert light, so the light that we see, to electrical signals. So those action potentials that are processed in our brain as seeing like vision. And this first takes place once the light hits the retina. So it's a pretty quick process. And then this is really fueled by those photoreceptors, those rods and cones, really are like the primary way that start this vision for us. And then one thing that's really mentioned in the textbook, as well as most of any lecture about any type of retinal layers, is convergence. So convergence is the process in which lots of neurons will synapse on one, so that allows for a lot of stimuli to be kind of analyzed at the same time. So here's a little bit more about the rods and cones. So they have those disc-like layers. The rods use rhodopsin, which is mentioned 2 slides before. And the cones are going to use 3 other types that are closely related to rhodopsin, but are more complex for the color vision. So they have 3 different ones for that. Rods have those 3 cation channels, and the CNG is going to be, it's called cyclic nucleotide gated, and basically those channels allow the sodium and calcium to enter, and rods also have the potassium leak channels and a voltage gated calcium, which helps with neurotransmitter release. And we remember from learning about action potentials and all that the sodium and calcium really are fueling the action potential and basically the potential for something to have an electrical activity. So the calcium is going to do that neurotransmitter release, which I'll talk about soon. So this is basically, in the next 2 little bullet points here, is more information about how they work. So in darkness, the cyclic GMP, which is basically a nucleotide as well, that allows the CNG and potassium to open, and then it's depolarized. So there's going to be continuous release of glutamate. Glutamate's a neurotransmitter that's an excitatory. In light, there's the rhodopsin as activated, and then the transducin is also activated. So here you can see, this is then I'll lead to a hyperpolarized state, and the glutamate will decrease. So here you can see an in-depth picture, which is really, really helpful to kind of visualize it. You can see how the mechanism in which it is going to be like opening and closing of the channels and how the membrane is hyperpolarized or depolarized and how the neurotransmitters are released. So this really helped me kind of solidify everything. And then this is just a little bit about like synaptic terminals and how they release onto them to the bipolar cells. So the bipolar cells can be on or off, and they are basically on when the glutamate decreases, and they're off when the glutamate is released. And then ganglion cells have two sections that are going to help visualize important information and strength of response, so more or no action potentials. Axons of the ganglion cells are going to make up the optic nerve. So the optic nerve is basically how all the information is transmitted to the central nervous system for processing. And that's going to make up my the rest of my presentation. And then here is going to be the structure of the retina. So this is my worksheet that I made. So you can see the structure of the retina. So here again is like that little square box, and then it's kind of zoomed in here to show the layers. This is like a labeling activity that will really help solidify like the way that information is kind of flowed. So you can see here the light comes down, and then you can label all those. And then here you're going to have a matching activity that has like basically the purpose of each cell and neuron and what every layer does for the retina. All right, and that's everything here. Thank you so much.